Okay, so programmatic access to UniProt knowledge base. To start with, since this webinar is only going to be an introduction to the topic, I'd like to start with a link to the technical section of the UniProt help section, which explains everything about the data structures that UniProt knowledge base provides, as well as how to access those data. The former is necessary to properly do the latter. The data structures are basically the file formats that we provide and the data and the structure of the data that are contained within those files, like, for example, FASTA headers or XML schemas. In terms of knowing how to access the data, the technical documentation explains the query fields that you can use to query UniProt knowledge base on the website, also explains all about the REST access, the UniProt J API and the UniProt Biomod, all of which I'm going to touch upon in this webinar in some fashion. When talking about programmatic access, most people think about accessing an online resource, i.e. querying the website live as it is. But of course, you can also download data in batch and look at them offline on your own local computer. Especially if you need a fixed data set that doesn't change and you just want it updated from one release to another, you might want to look into this. There is a download section on the UniProt website, which is called uniprot.org/downloads, where you find many pre-computed data sets that might actually satisfy your needs. In terms of, with regard to UniProt knowledge base, you'll find the entirety of SwissProt downloadable, both in text and XML format. If you're interested in sequences only, you can download FASTA. You can download sequences only, including isoforms. The same is true for Tremble entries. And you can download various additional data sets like taxonomic divisions and the like, and also other resources like UniRefs and UniPark are available. So in case you need these data sets, go look them up and download them, and you can analyze them locally on your machine. But programmatic access, sensu stricto, is more about accessing the live data as they're presented on the website. And we're going to touch upon the UniProt REST interface, accessing those services also via the EBI web services, using a Biomod and the UniProt J API, although the focus is going to be on the item number 1, the UniProt REST interface. What is REST? REST is obviously an acronym for a very technical term. In layman's terms, it describes a way to interact with a resource, in this case UniProt, and to retrieve data from that resource. I'm not a programmer either, so the technical detail will be kept to a minimum in this webinar. The nice thing about REST is that it can be used with any programming language, and since it is about interacting with an online resource, it is URL based. Since URLs are normally not very nice to look at, it helps to break them down into components to understand how they're constructed. With regard to REST, you have basically 3 components in such a URL, which you can see on the slide here. You always have a provider of the data, which in this case is uniprot.org. This provider can provide one or many resources. Within UniProt, we have one paramount one, which is the knowledge base. The endpoint is here, UniProt. And then you just append the data you'd like to retrieve. And the URL that's given here as an example, you can actually copy and paste into your browser, and you will see the result. And this is a REST interaction, even without any programmatic access as such. The UniProt REST interface has various resources that are exposed. I've already mentioned the UniProt knowledge base up at the top here, but we also have a mapping service for identifiers, which is exposed with the mapping name here as the resource. This allows you to map, for example, a UniProt accession to an EMBL identifier and vice versa. There's also a batch entry retrieval service if you don't actually want to launch a particular query, but you know about a set of entries with their accession numbers that you'd like to retrieve. You can do this using this endpoint of the REST interface. You can also access UniProt reference clusters, basically clusters, protein sequences that lump together protein sequences based on the sequence identity. And finally, there's also the UniProt archive, which allows you access to the archived versions of sequences and see how sequences change over time. We're going to focus in this webinar on the first one of these resources, which is uniprot.org/UniProt, the UniProt knowledge base REST interface. The parameters that you can pass to such resources are listed here just for UniProt knowledge base. Obviously, they depend on the particular resource, and you'd have to look them up in the technical documentation. The most important parameters for the UniProt knowledge base REST interface is first and foremost the query parameter that allows you to pass a query like taxonomy.9606 and reviewDS, which translates into plain English as all human entries in SwissProt. You can specify a format. Again, the selection of formats available depends on the resource you're trying to access. For UniProt knowledge base, you can specify, for example, FASTA if you're interested in sequences, .txt if you're interested in the plain text version of the entries, XML if you're interested in the XML version, or tab delimited format. That's not all. There are some more that are not listed here. If you opt for the tab delimited format, there's an additional parameter you can use, which is called columns, and this allows you to specify which fields you'd like to retrieve. More on that a little bit later. Another parameter is the limit parameter, which allows you to specify a maximum number of entries to retrieve based on a query. This is particularly handy if you don't know how many entries to expect when launching a query, and you're afraid that it might actually be so many that it brings down your network connection or it just takes too long. Okay, all of this sounds very complicated. The good news is you don't actually have to learn all of these parameters by rote just by looking at the technical documentation. Instead, you can explore them using the interactive advanced search on the uniprot.org website and have the URL constructed for you. The way this works is like so. On the UniProt website, you can go to the search box and click on the Advanced tab, which opens up a view like this, where you can, in a point-and-click fashion, construct very complex searches that target individual fields with the information that you're looking for. And once you click Search, this is translated into a search string, as you can see here. And also, once you click the search button here, it is also translated into a URL, which is displayed in the URL bar of your browser. And this is basically the REST URL as you would want to construct it when doing programmatic access. Now, with this, you're already set to go to actually use programmatic access to the UniProt knowledge base. If you're on a Unix-like system, you probably have tools like curl available, which is just a command line tool that accepts a URL like the one we've just looked at, downloads the results hiding behind this URL, in this case saves it in a file called my_results.txt. You could specify anything, obviously, what you want. For very simple queries, you could even construct the URL yourself. For example, if you're only interested in a particular UniProt entry with the accession number P12345, you can write it like so and download the entire entry in just this one line. But obviously, having a URL like this one is not very flexible. This construct once, use often approach is limited. It's not very dynamic. As soon as the query changes, you either have to go through the steps of reconstructing the search URL using the advanced query interface, or you have to start fiddling around with the URL, which is not very nice. At this point, you should consider using a programming language. For the sake of simplicity, I'm going to use Python as an example of a programming language for this. First of all, because I know a bit of Python, and also because the code is fairly easy to read even if you have no prior programming experience. This here, for example, I'm going back to the example that we used before, where we searched for a gene, a particular gene name, tax1, BP1, and HTLV, which happens to be a particular virus that this gene occurs in. If we reuse this example, you could construct a search function which looks very intimidating at first look, but it holds all the components that we already know about that are part of a REST URL. We have the provider, which is specified here. We also have UniProt again. We have the query parameter, which happens to be exactly the same like what you would type in the search bar on the UniProt website. Then we can specify an additional parameter, the format parameter, which I've already mentioned, which in this case would default to txt, download the plain text version of any entries that a query retrieves. So with this, I think it becomes immediately more easy to understand how such a construct is built. Obviously, the advantages of such an approach is that it's very flexible. You can provide different queries and very many queries to the same function. You can specify different formats and see what are the differences in terms of download. And such a function is also reusable. You can use it in many different ways and different pipelines that you can tie together and integrate with one another. The disadvantage, obviously, is you have to write the code. Well, it turns out that you don't have to write all of it. Because, as happens so often, many other people have been through the same experience than you have and faced the same problems, and some of them have used all their efforts and packaged them up so that they're reusable for others. One of such examples is the Bioservices package. It is a Python package which was developed by people at EBI, and it provides easy access to many bioinformatics web services, UniProt among them, but it's certainly not limited to UniProt alone. Check it out if you're interested. With using this package, the code that we looked at just a minute ago, I'm going to go back. This code basically boils down to just 4 lines of code, but the components are still there. They're just clearer and easier to see, I think. So we have the provider, which again is UniProt, here boxed in black. We also have the resource, which in this case, boxed in green here, is search, which means we'd like to search UniProt knowledge base. We still pass a query parameter, which happens to be the same as in the previous example, and we still specify a format parameter, and it's all done. And once we've retrieved the results and print them, this is what we'd get, just what we'd expect, the plain text version of the query results. In another example, this demonstrates the flexibility of this approach, where we basically still have the same provider, we still search UniProt knowledge base, we provide the same query parameters, but choose a different format now. We go for the tabular format, and we combine this with an extra parameter called columns, where we specify only the bits of the entries that we're actually interested in. In this case, the entry name, the length of the sequence, the ID, and any gene names that might be present in such an entry. And if we do this, this is what we'd get. We get a tab-separated result where just these pieces of data are present. At this point, I'd like to make a 2-slide detour and talk about the columns because I think they're very useful when starting to think about programmatic access. There is, in the UniProt help section, a very comprehensive list of all the column names that you can possibly want and that are available when dealing with UniProt knowledge base entries. And they're to be used in combination with the tabular format to specify, like I said, only those pieces of information you're interested in. It is very powerful, as the downloaded information can be kept to a minimum. just imagine that if you launch a query that returns 1,000 hits and you download the full text versions, you'd actually have to download 1,000 complete entries, including the references, etc., etc., even if you're just interested in, say, the keywords contained in those entries. So you would download an awful lot of information, which you afterwards just discard and waste bandwidth on. Instead, if you just choose tabular format and specify keywords as the columns, that's all you're downloading. So it's very efficient. Okay, an example to illustrate this is if you were to search for all reviewed human proteins which have a topological domain intramembrane annotated, and you would only like to retrieve the relevant bit of information, which in this case happens to be the intramembrane bit, you could construct a query. It's also linked here and it takes you to the website like this, and it would give you the results as displayed at the bottom of the slide here. So since this would target all the human entries in not just SwissProt, but possibly all UniProt, that'd be an awful lot of entries to download if you didn't choose the tabular format. So this shows the power of the tabular format. Now on to a different way of accessing the UniProt REST services, which is via the EBI web services. There is an EBI-wide search which is called the EBI, and it allows an EBI-wide search of databases. It's not limited to UniProt, and the access is also via REST, so it follows the same principle. There used to be also a SOAP interface if someone has come across this, but this will be phased out towards the end of 2015, so REST is the access to go for. There are sample clients available, for example in Python, but also other languages like Perl. The formats you can retrieve data in are various. It usually defaults to XML. Often JSON is available, which is a type of format that is not available using the UniProt REST API as such. I've put a link on this slide that guides you to a page that lists all the indexed fields that you can retrieve and search using this access to the UniProt data. It is a little bit different from using the UniProt's own RESTful API. There's also another service called WSDB Fetch, which if you don't actually want to run a query but instead know about entries you'd like to download, you can do this using this service. EBI-wide search follows the same principle as the UniProt REST interface, but of course there's a different provider. In this case, the URL is specified as shown here, and there's just the one resource which is UniProt, and it targets the UniProt knowledge base. And the syntax for parameters is slightly different. For example, taxonomy has to be spelled in uppercase, and the field names are different from what you would find in the UniProt RESTful API, but that's documented and you can look it up. I've already talked about this. An example, for example, if you searched for human entries and the word antigen, and you would like to retrieve names and the scientific name of the organism in the JSON format, this is the URL you would construct. And down here, in the bottom half of the slide, you see the results you would get. One nice thing about this approach is that it always tells you about the number of hits, even though by default it only retrieves 100 or so. So you'll never run into the trouble of actually downloading an awful lot of data, even if your query returns quite a lot of entries. Another example where you actually don't want to search UniProt knowledge base as a whole is, but instead would like to retrieve particular entries. This is possible by appending yet another resource to the UniProt one in the provider, and this is entry. Once you've done this, you can provide either a UniProt accession or a UniProt ID, as is shown here, and retrieve specific information for just the list of identifiers that you provide in the URL here, which in this case are just 2 human proteins. You need to get something like this one down here. You have OMIM numbers which are retrieved as part of the data. The advantages of the EBI web services for querying the UniProt RESTful API is that once you're familiar with it, it's probably very easy to transfer your knowledge to other resources hosted at the EBI, which makes it more powerful in this sense. And JSON is a return format which is available, which is not the UniProt REST API. Disadvantages? As far as I know, there are no such nice packages like the bioservices one for using it, at least currently. There are sample clients provided though, and not all the UniProt data are accessible. The comment section as such is not indexed right now, and I think it's mostly about cross-references, taxonomy, and the like that you can download. Okay. The third mode of accessing UniProt data programmatically is via Biomart. Again, there's a link to the actual resource at the top of the slide. It allows you to point and click in order to build your query. There are 3 basic steps. You have to choose a data set. For the UniProt Biomart, there's only one data set available. It's UniProt. And then you have to choose filters, which is the equivalent to specifying a query. So you either provide a list of accessions, which then is sort of like a batch retrieval of entries, or you provide a species name or protein name, etc., and launch a query. You specify the fields or the columns you'd like to download in the so-called attributes section, where you can specify, for example, that you'd like to download Go terms or EMBL IDs for all the search hits that you get. The formats that are available for result viewing are HTML, XML, CSV, and TSV, so fairly limited. Queries can afterwards be saved as an auto-generated Perl script or a URL, so they're easy to rerun and reuse in a static sense. So unless you'd like to fiddle with those, you'd have to reconstruct any changes to your query by pointing and clicking the interface of the Biomart. So there's also a disadvantage in the limited range of attributes that are available for retrieval. What it looks like is this. This is the Biomart interface. On the left-hand side, you have the filters. There's already one selected, which in this case means only those that belong to the complete proteome of Homo sapiens. There's a selection of attributes already specified. And once you retrieve the results based on this, you get a view like this. It's the HTML display. You could choose different ones. And up here in the gray bar on the right-hand side, these buttons would allow you to either download and save the URL, the query in XML format, or as a Perl script for later use. Like I said, Biomart has its limitations. I think the RESTful API is more powerful. Finally, there's the UniProt J API. It's available for download from the link on the very top. I'd like to point out that there is a much improved version which is currently in beta, available from the URL here in the middle. If you'd like to use it, I think it requires Java 8. If you are using it, and we would be happy about hearing feedback, since we're still improving things and ironing out bugs. So please do contact us if you have anything to share about this. The UniProt J API provides the most comprehensive access to UniProt data. It's also used internally for handling a lot of the data. It allows you to search UniProt as well as UniPark and UniRef, just like the RESTful API, but you can also run blast searches. The disadvantage compared to the RESTful API is that it requires just one programming language, which is Java, and this is not lightweight like the approach that we've seen before, but rather wordy. Down here in the black box is just a snippet of code that would not get you anywhere. Many more lines of code are required to just retrieve a single entry. So this is more for larger systems and applications, and not just lightweight access for exploratory data analysis, for example. Okay, now what we've talked about so far is that the REST API allows you to, or all the other means of accessing UniProt data programmatically allow you to run queries and download the data. Now, the UniProt J API provides access to all bits of a UniProt entry. It represents it as like a virtual object that you can poke and explore. The other means, for example, if you're retrieving entire entries in text format via the REST API, afterwards require you going over the entries and retrieving the bits of annotation that you're actually interested in. That is, unless you use the tabular format with just the bits of information, the columns that you need. If you opt for downloading entire entries, be it in text format or, for example, XML format, you have to parse them afterwards. You have to read them in with another program and filter out the bits that you're interested in. With regards to Python, there is a package for this. It's called BioPython. It allows you to parse both the text version of SwissProt or UniProt entries as well as the XML versions and iterate over entries and extract just the bits of information that you want. There are similar libraries for Perl, for example. I think it's called Swiss Knife, and possibly others for other languages which I'm not familiar with. So this was the general introduction to programmatic access to the UniProt knowledge base. If you're stuck in any way, not just with respect to programmatic access, just ask. We have an active help and support team. Contact help@uniprot.org. And also, if you find something incorrect, outdated, or missing, please let us know. We're always happy to help.